Beneath the ice-framed water of the Antarctic Peninsula, the seafloor does not look like a pharmacy. It looks dark, cold, and almost impossibly remote. Yet attached to the rock is a small, soft-bodied animal whose chemistry has drawn cancer researchers back across the Southern Ocean. It is called Synoicum adareanum. It is an ascidian, better known as a sea squirt. It has no claws, no teeth, and no dramatic chase scene. It spends its adult life fixed in place, filtering seawater through a body that can resemble a pale, uneven cluster on the ocean floor. But the most important creature in this story may be living inside it. Within the sea squirt is a community of microbes. One of them carries genetic machinery linked to a molecule called Palmerolide A. In laboratory studies, that molecule showed unusually potent and selective activity against melanoma cells. That does not mean Antarctica has produced a melanoma cure. Palmerolide A is an experimental research compound. The evidence discussed here is preclinical, and the cited sources do not establish that it is safe or effective in humans. The real discovery is more careful and perhaps more fascinating. A stationary animal, a bacterial partner, and a chemical defense built in one of Earth's most isolated seas may teach science how to search for future medicines without stripping the wild place where the clue was found. This story begins near Andros Island on the Antarctic Peninsula. The water is cold enough to remain close to freezing, and for much of the year the world above it is shaped by ice, wind, and darkness. Below, life grows on rock faces, slopes, and vertical walls swept by currents. Sea squirts are filter feeders. Water enters through one opening, suspended food is captured, and filtered water leaves through another. The name may sound comic, but ascidians are sophisticated marine animals and surprisingly close relatives of vertebrates within the chordate family tree. Synoicum adareanum forms colonies. Many individual units live together within shed tissue. To a diver, the colony may appear modest beside a starfish or a brightly colored sponge. To a chemist, however, it can contain an entire landscape of molecules. Animals that cannot run from predators often rely on other strategies. Some hide, some build armor, some form partnerships with microbes, and some use chemistry to make themselves less appealing to whatever might bite, infect, or overgrow them. In the Antarctic, survival can be written as a molecule. In 2006, researchers reported the isolation of Palmerolide from Synoicum adareanum collected near Palmer Station. Chemically, it is a complex macrolide, a large ring-shaped natural product. Biologically, it immediately stood out. In a cancer cell screening system, Palmerolide A showed potent activity against a melanoma cell line. The original study reported a lethal concentration of 18 nanomolar for the tested cells. At that scale, a nanomolar is 1/1000th of a mole per liter. Potency in a cell line is an early signal, not a clinical result. A compound can look extraordinary in a dish and still fail because it cannot be produced, delivered, tolerated, or controlled inside a living body. What made Palmerolide A especially interesting was not simply that it killed cells. Many toxic substances can do that. The attraction was selectivity. In the original screening profile, melanoma cells appeared much more vulnerable than many other tested cell types. Researchers also linked its activity to VAT-PACE, a molecular machine that helps control acidity inside cellular compartments. These proton pumps move hydrogen ions across membranes. Disrupting them can interfere with processes a cell needs to survive. That mechanism is powerful, which is exactly why caution matters. A useful medicine must strike the right biological target at the right dose, in the right tissue, while limiting damage elsewhere. Discovery begins with an effect. Drug development begins with all the reasons that effect might not be usable. There was another mystery. Scientists had isolated Palmerolide from the sea squirt, but that did not prove the animal itself manufactured the molecule. Marine invertebrates often live with dense microbial communities, and some famous animal compounds are actually made by bacterial partners. The sea squirt was not a container. It was an ecosystem. Researchers began examining its microbiome, the bacteria living within its tissues. They also searched for biosynthetic gene clusters, groups of genes that work together like a molecular assembly line. If the right cluster could be found, it might reveal who was building Palmerolide A. That search was unusually difficult. The suspected producer could not simply be grown in an ordinary laboratory dish. So the team used metagenomics, sequencing genetic material from the entire microbial community and then reconstructing genomes from the mixture. In research published in 2021, scientists identified a 74 kilobase candidate biosynthetic gene cluster containing machinery consistent with Palmerolide production. The cluster was associated with previously uncharacterized bacterium in the Ascidians core microbiome. They proposed a name, Candidatus Synoicum Palmerolidicus. Candidatus signals that the organism has been characterized through genetic evidence but has not yet been fully established in culture under the conventions used for formally described bacteria. The reconstructed genome contained not one, but 5 related copies of candidate Palmerolide biosynthetic gene clusters. That repetition suggested this chemistry was not an accidental trace. It may be an important part of the bacterium's life with its host. The word candidate remains essential. The genetic machinery strongly supports the link, but science still asks how the pathway operates, what each gene contributes, and how the final molecule moves through the partnership. The ecological question is just as compelling as the medical one. Does the bacterium make Palmerolide A to defend itself? Does the compound protect the sea squirt from predators, infection, or competing organisms? Does the host feed the microbe, shelter it, or influence how much chemistry it produces? These are not side questions. If researchers hope to reproduce the molecule without collecting wild animals, they need to understand the factory, not merely possess the product. The bacterium's genes, metabolism, and relationship with its host may all matter. A spatial survey published in 2020 offered another clue. Scientists sampled 21 ascidians across 7 sites around the Anvers Island archipelago. Palmerolide A was found throughout those samples, and the animals shared a core set of bacterial partners. That consistency made the relationship look stable across the region studied. But Antarctica is vast, and a cluster of sites near one archipelago cannot answer every question about distribution. How far does the ascidian extend? Where does its bacterial partner occur? How do depth, current, temperature, and season change the partnership? To move forward, the scientists had to return to the ice. In 2026, a National Science Foundation-supported team spent 6 weeks in Antarctica. According to the University of South Florida, the expedition focused on locating the ascidian's Palmerolide-linked bacterium, mapping how widespread it is, and understanding how it lives within the host. The team searched seafloor habitat along the Antarctic peninsula. Divers collected samples, while 2 remotely operated vehicles helped explore deeper water and identify additional sites. This was not a treasure hunt for a miracle substance. It was field ecology connected to chemistry and genomics. The ascidians were commonly found around 60 to 80 feet deep, often on sloped or vertical surfaces where currents were strong. Those currents matter. A filter feeder depends on moving water to deliver suspended food, oxygen, and the conditions that sustain the colony. Working there compresses science into short windows. The USF team reported average dives of roughly 25 to 35 minutes, with a maximum depth of 130 feet. Every descent had to account for cold, ice, changing seas, visibility, and the presence of leopard seals. A sample brought to the surface is not just a piece of tissue. It is a chemical moment. Poor handling could alter the organisms, the bacteria, or the compounds researchers hope to measure. Collection, preservation, and transport become part of the experiment. Back on land, the new material enters a much slower phase. Teams examine DNA chemistry, biological activity, and geographic patterns. The university cautions that this analysis can take months to years. The expedition is a new chapter, not a finished answer. There is no human clinical trial result in this story. There is no approved Palmerolide medicine. The current achievement is a better map of a natural system and a stronger path toward testing whether its chemistry can be reproduced and developed responsibly. That production problem is the hinge of the entire project. Even a promising molecule is of little use if every dose requires removing rare animals from an Antarctic ecosystem. Wild harvesting would be difficult, variable, ecologically damaging, and impossible to scale responsibly. The Antarctic Treaty System also protects the continent for peaceful and scientific purposes. The goal of research cannot be to turn a protected seafloor into a pharmaceutical quarry. The better route is to learn the biological instructions and rebuild the process elsewhere. That could mean placing the necessary genes into a laboratory organism that can produce the compound. It could mean cultivating the original bacterium under conditions scientists have not yet mastered. It could mean total chemical synthesis, semi-synthesis, or a modified version that is easier to make and safer to test. Each route has trade-offs. Total synthesis gives chemists control, but may require many difficult steps. Fermentation can be scalable, but only if the biological machinery works in culture. Engineering a host organism demands a precise understanding of a long and complex gene cluster. The Baker lab at the University of South Florida received a National Institutes of Health grant for a 5-year effort that includes developing ways to make promising anti-melanoma compounds from Antarctic ascidians in a laboratory rather than relying on Antarctic collection. The most valuable thing brought home may not be the ammo. It may be the recipe. Nature-derived drug discovery often follows this pattern. A plant, fungus, sponge, tunicate, or microbe produces a molecule for reasons that have nothing to do with human medicine. Evolution selects chemistry for survival. Researchers notice that the same chemistry affects a disease-relevant process, but nature does not design medicines for us. A defensive toxin may be too dangerous. A molecule may break down too quickly. It may never reach the right tissue. It may affect a molecular pump found in healthy cells as well as cancer cells. Every promising effect opens a longer list of tests. Melanoma itself is not one simple target. It begins in melanocytes, the cells that produce pigment, and it can invade nearby tissue or spread to distant parts of the body. Current treatment can include surgery, immunotherapy, targeted therapy, radiation, and other approaches depending on stage and tumor biology. Modern melanoma care has changed dramatically, yet the National Cancer Institute notes that many people still do not benefit from newer drugs, and others relapse after an initial response. New mechanisms remain worth investigating, but they must pass the same rigorous path as every other treatment candidate. If you are looking for medical guidance, this documentary is not a substitute for a qualified clinician. Its subject is how a natural product lead enters research, not how any person should treat cancer. For CreatureCosmos, the deeper wonder is the partnership. Synoicum adareanum is visible. The bacterium is microscopic. The molecule is smaller still, yet the three are linked across scales. Habitat shapes the host shelters a microbiome, and microbial genes may build chemistry that changes how the whole colony survives. Remove one layer, and the story becomes misleading. Call the ascidian a miracle animal, and the bacterium disappears. Call the molecule a cure, and decades of careful development disappear. Call Antarctica an untapped resource, and the living ecosystem disappears. The accurate version is better. The animal is a habitat. The microbe may be a chemist. The molecule is a clue, and the scientific task is to understand all three well enough to protect the original system while testing whether its chemistry can help people. There's also a lesson for conservation. Species do not only represent bodies counted in a survey. They hold relationships, behaviors, genomes, and chemical pathways that may exist nowhere else in the same form. Losing an organism can mean losing an entire network before we learn its language. Antarctic benthic animals evolved in prolonged isolation under cold, seasonal, and chemically demanding conditions. Reviews of Antarctic marine invertebrates describe a wide range of bioactive natural products from sponges, cnidarians, mollusks, bryozoans, worms, echinoderms, and tunicates. Most will never become medicines. That is normal. Drug discovery is a funnel. Many compounds show an interesting effect, fewer survive laboratory validation, fewer still reach animal studies, and only a small fraction enter and complete human trials. Failure does not make the exploration pointless. Even when a candidate does not become a drug, it can reveal a biological mechanism, inspire a new synthetic method, identify a microbial lineage, or show how two species cooperate. Palmerolide research has already connected natural product chemistry, polar diving, microbial genomics, ecology, and cancer biology. The 2026 expedition adds the missing geography. Where does this partnership occur? Is the bacterium present in every colony? Does palmerolide concentration change across depth and habitat? Can researchers link the molecule's abundance to the health or ecology of the host? Those answers will decide what comes next. If the biosynthetic pathway can be reconstructed, scientists may produce enough material for more complete testing. If the compound's selectivity holds under increasingly realistic conditions, it may justify further development. If it fails, the data will still sharpen the search. That is what responsible wonder looks like. It leaves room for astonishment without skipping evidence. It can celebrate a molecule that kills melanoma cells in a laboratory while stating clearly that a cell result is not a patient result. It can recognize the urgency of cancer research without promising a cure from the ocean. And it can value Antarctica not because everything alive there must be useful to us, but because life has assembled solutions there that we have barely begun to understand. On the seafloor, Synoicum adareanum continues to filter cold water. Its bacterial partner continues an invisible metabolism. Palmerolide A continues to exist for an ecological reason that may have nothing to do with melanoma. Science did not find a medicine cabinet under the ice. It found a relationship. Whether that relationship ever leads to a treatment will depend on years of chemistry, biology, safety testing, and clinical evidence. But the path is now clearer. Identify the producer, understand the genes, recreate the chemistry, and leave the Antarctic colony where it belongs. The creature that might teach medicine a new trick is not acting alone. It is a colony within a community, carrying a microbial factory in a frozen sea. And its greatest lesson may be that the future of discovery lies not in harvesting nature, but in learning how nature builds. If you want more source-aware stories about real creatures that feel stranger than fiction, subscribe to CreatureCosmos. And tell us in the comments, which hidden partnership should we investigate next?